So let me just show you a couple of examples. So if you look at the probability of going from phase 1 all the way to completion, that's this column right over here, phase 1 to completion. With this study, you're getting about 13.8% probability. That's across all drugs, all indications, all of the analysis in our results. If you take a look at Thomas et al., 2016, they're getting a figure of about 9.6%. Hay et al. Is 10.4. Another paper, they get 15, and then Damasi gets 19. So you can see a lot of variability across the different papers in the literature. We obviously have more confidence in our results. We haven't bothered calculating standard errors, but if we did, the standard errors around our figures would be really tiny because we've got huge samples to calculate this from. So that's one interesting statistic. But in addition, you can see that there's a lot of variability from phase 1 to phase 2, phase 2 to phase 3, and so on. So for example, phase 1 to phase 2 is about 66%, phase 2 to phase 3 is 58%, and then phase 3 to approval is about 59%. Now, one of the interesting things about this is phase 2 to phase 3 of 58%. And let me just explain to you what that means. That means that a phase 2 successfully transitions to phase 3. And then if you want to know what is the chances that a phase 2 successfully transitions all the way to an approved drug, that's in this column. That's about 35%. So I've talked with some pharma executives about that, and there's a very interesting perspective. The perspective that they take is that if you start with a successful phase 2 drug, you've got phase 2 successfully completed, and you've hit your endpoints, there's no excuse for why a phase 3 should fail. And the reason for that is simple. Phase 2 is a smaller sample of patients. You're testing a very specific set of things, and you get a result that presumably is statistically significant in that small sample. If now all you do is to increase the sample, if you increase the sample exactly as you specified it in phase 2, you should be getting an even stronger result in phase 3. So the claim is that the reason that people fail in phase 3 when they succeeded in phase 2 is because they didn't obey that very simple axiom, which is keep everything the same other than the sample size. Now, in some cases, you can't, because, for example, in phase 2, suppose that you got good results, but you also got some toxicity, so that a couple of patients died, even though the patients that survived actually had some significant benefit. What do you do then? Well, maybe you lower the dosage. If you lower the dosage and you enter a phase 3 trial, you're just asking for failure, because you didn't test a lower dosage in phase 2. You tested a higher dosage, and yes, it had toxic effects, but now when you do a phase 3, you've just changed the design. So the right answer is you need to do another phase 2 study with a lower dose. But it's very hard sometimes to do that for two reasons. One is time is money, and your career is ticking, so you want to see a drug that you shepherded through the process to completion. You want to get going. And the second reason is actually a perfectly laudable one. It has to do with ethics. This is a good drug. You just showed it was a good drug. People can't use the drug until it gets FDA approved. So there are people that may be dying from a particularly desperate disease, and you've slowed down the process by doing another phase 2 study. So it's very difficult. Human emotion plays a huge role in this. I'm trying to emphasize behavior here. So this number, 35%, may seem shocking to a lot of people. It's because it's an example of a phase 3 study that somehow has changed in design from phase 2. So just keep that in mind when you start designing these things yourself.